Uh, I just got in here from Los Angeles. My flight was supposed to arrive last night at 1. And then somebody had a heart attack on the plane. So the plane got diverted to Toronto. Then I missed my connecting flight, so I landed in Dubai. So if I pass out now after 30 hours of flying, it's not my consciousness. It's just the lack of oxygen. It's a pleasure to be here despite the gruel travel. I'm going to be talking about stuff that is quite different. And I'm going to be defining a whole lot of things. As the previous speaker said, cognitive science is the new kid on the block. In terms of that, neurophysics is the new infant on the block, perhaps not even. So this is what neurophysics means for me. Neurophysics is mathematically sound theories of the brain that are rooted in biology and are experimentally and quantitatively falsifiable. So all these are requirements. If one of them is not met, then it's not really neurophysics. And this is not something I'm making up. That's how physics has been. It was not just introspection. Introspection didn't really take us too far in physics. It was experimental falsifiability of mathematically sound ideas. That's what led us to lasers, which nobody would have imagined, perhaps. So with that, I want to introduce my biases and working hypothesis. I must say off the chart, I have no idea what consciousness is. I meditate. I have certain feelings when I meditate, but I don't know what that is. I'm working on it, along with many people in this style of work. So these are biases and working hypothesis, and I'll have a little bit of a theory. It's still the beginning. So bias number 1, humans are a part of a genetic continuum, and hence most animals are likely to be conscious. These are, if you wish, these are axioms. I'm not going to argue about it. This is what I'm going to start with. Second, consciousness is something to do with the brain. I don't know what it is, but something. May have more than brain, but brain definitely. Number 3, brain organization varies considerably between species, from invertebrates to vertebrates. There is no anterior singular cortex in invertebrates, for example, something that we know and love in mammals. Hence, consciousness is unlikely to reside in a specific brain region, and the underlying mechanisms must be stable across evolution, because otherwise nature would have to reinvent consciousness with each step of evolution. So we must have some invariant mechanism that goes back millions of years. So it's a big challenge. Not only that, the brain size varies by a factor of 100,000 or 1,000, even within mammals. Compare the size of the brain of a mouse versus an elephant. And the brain size changes with development substantially, with connectivity changing. We have 100 billion neurons, but the connectivity changes substantially from several trillion to not even a trillion with development, and large is not necessarily the best. Hence, consciousness mechanisms must be scale invariant but scalable at the same time. The same mechanism of consciousness should apply to a mouse as to an elephant, and this is the only time I'm going to be religious. Think of Ganesha. That's where these two come together. That's a joke. But both of them are supposedly conscious, but evolutionarily quite far apart. So the same mechanism must apply to all of them. Otherwise, we'll have to think of too many mechanisms, and then I don't know what we are talking about. Consciousness exists in the absence of external stimuli. We all know that, at least through introspection. It doesn't go away, as somebody has mentioned. For example, in darkness or in silence, different modalities are taken out. Hence, mechanisms of consciousness should work in the absence of stimuli or behavior. So based on this, this is the bedrock of the minimal set of parameters or observations that consciousness theories must satisfy. Mechanisms should be scale invariant but scalable. They should be stable across behaviors in time and evolution. Time in terms of development in person's life, and evolution in terms of much longer time scale. Semantics. What I'm going to describe later may not necessarily be consciousness, maybe awareness, maybe attention. I don't really know what those are. If we can figure out even one of them, I think we'll be very happy. The way we want to start in neurophysics is way human beings started, with a pendulum or a simple harmonic oscillator in physics. We were thinking about the universe. There are lots of theories of the universe in the Christian and the Vedic literature. But the progress was made when human beings decided, let's forget about those things for a little while, and let's think of a letter that doesn't even exist. at that time at least. When people solved it, progress was made, and now we can talk of things of how the universe began. So I'm going to talk about something which is equivalent of simple harmonic oscillator in regards to consciousness. And I want to do it in an experimentally testable way. So one way I want to do it in an experimentally testable way and in an objective way is to take out introspection. I want to find a way that I can manipulate consciousness experimentally. And the manipulation is in my hands. And I can do that in two very distinct ways. One is through anesthesia. I can control the level of anesthesia, as I will show you in experiments we do. Or the other is to make somebody run, kind of the opposite. You're very unlikely to be asleep when you're running. Very few people can do that. I might. Right now I may be talking while I'm asleep, but that's a different story. So these are the two parameters I can control. I can measure, along with it, I can measure physiological parameters, and I can objectively tell you what might be going on. So the first question becomes, what does the brain do when we are asleep or anesthetized? Isn't it remarkable that we all need to sleep? Otherwise we are dead. It doesn't take energy to consume, but still we need to sleep. Mysterious. So why is it that all animals need to sleep? I won't be able to answer that. That's too long a story. We are going to zoom in now. All the philosophy or biases are over. We are going to start with the science now. So the first surprise is that the brain is not silent, even when we are anesthetized. Let alone when I lie down, close my eyes, turn off all the lights. My visual cortex, responsible for seeing colors and lights and so on, is as active, more or less, 10 or 20% difference, than when I'm paying full attention to what's on the screen. Very small difference. So the brain is remarkably active in terms of metabolic cost, in terms of the number of spikes fired by neurons and so on. So first thing is, why is it so active? Maybe that activity of the brain in the absence of any stimuli or action has something to do with consciousness. As pointed out by the previous speaker, maybe that's the default network. But it's all over. So this is a neuron that we have recorded. This is a beautiful neuron. Notice this is 250 micrometers, the scale bar. This is the first recording done from these neurons. These are the neurons in the so-called posterior parietal cortex. There were beautiful experiments done on out-of-body experience. Yes, there were scientific experiments done 6 years ago in Switzerland. When a woman had a serious stroke and they had to implant electrodes in the posterior parietal cortex, whenever they passed a small amount of current in the posterior parietal cortex, the woman had a reliable out-of-body experience. She saw herself from the top of the ceiling. That was subjectivity, but that's as close as we can get to some manipulations of consciousness. So we focused on what goes on in posterior parietal cortex. That's one neuron. There are 10,000 synapses on this. We are not going to look at it. And we are going to look at what that neuron does when the mouse is anesthetized. This is time in seconds. That is the amplitude of the signal in z-scored units. So we know how significant the results are. Before 2.5 standard deviation, we cannot talk. That's the signal in millivolts. In blue, you are seeing is the average activity of the brain in that entire patch of the brain, measured through something like EEG called local field potential. And in red is the membrane potential of this neuron, measured right there at that spot in a 10 micrometer resolution. And what you see is that both the local field potential, which is the aggregate activity of the entire region, and the membrane of that individual neuron are showing you synchronous oscillation. They are pretty much coherent. Correlation is 0.9, which is pretty huge in biology given that all kinds of stuff is going on. They are both showing you these synchronized oscillations, and I'm going to focus on this synchronized oscillation for the rest of the talk. These oscillations not only occur in parietal cortex, but all sorts of brain regions, and the neocortex during that period behaves like a two-state system, switching synchronously between the up and the down states. And this happens during all sorts of states, not just anesthesia, during quiet wakefulness, as well as during slow wave sleep. This is what the most common thing your brain does. About 40% of your life is spent in this state. Up and down state means that the whole system becomes quiet, very inactive, and then suddenly it becomes quiet, the transition from inactive to active, that whole period is approximately 1 second long. The frequency is approximately 1 hertz, that 1 times a second in all species. So there is the first surprise. Whether you measure this signal in a mouse or a human brain, a factor of 100,000 difference in brain size, the frequency of the rhythm is identical. Identical. And there are a couple of other interesting features. The up states are more stochastic than down states. You can just see it without me quantifying it. Look at the up state. There's a lot of noisiness. The down state is much more quiet. Not only that, the up transitions are faster than down transitions. From here to here, the system takes much shorter time than from there to there. That transition is gradual. So I can quantify this mathematically, but these are the features that we have extracted in what the brain does in all sorts of species people have investigated. The latest paper that came out last year, they measured this in Drosophila, a very difficult experiment, a fruit fly, much smaller than the entire brain of the mouse, same frequency of oscillation. Virtually all parts of the brain in all species have roughly 2 types of neurons. So now I'm going to start making a theory of this simple slow oscillation. It's called the slow oscillation. There's ancient literature going back 100 years about it. 1920s is when the first measurement of these slow oscillations were made. It's that ancient. Now we look into the brain and try to make a theory that satisfies this huge list of demands that are made on myself and on everybody else. One thing we know, there are many types of brain regions, there are many kinds of activity patterns and so on, but one common thing is that in virtually all parts of the brain, barring 1 or 2 small exceptions, whether it's hypothalamus or neocortex or hippocampus or declarative system or non-declarative or prefrontal, there are basically 2 types of neurons, excitatory neurons and inhibitory neurons. Excitatory neuron, in the simplest term, once you activate that neuron, will excite whoever it's connected to. It will make them more active. Inhibitory neurons are the opposite. Once you make them active, they'll make the other neurons less active to whomever they are connected. And the wonderful thing is that whenever these excitatory and inhibitory neurons exist, they are connected to each other. This is one invariant feature starting from the invertebrates all the way to us. So not a bad thing to focus on. So now we are going to make a circuit of 2 neurons. 1 is not a circuit, 2 is a circuit. And we are going to focus on this mean field theory. So here is one excitatory neuron, here is this inhibitory neuron. The excitatory neuron can connect to itself, the inhibitory neuron can connect to itself, the excitatory neuron connects to inhibitory neuron, and the inhibitory neuron connects to excitatory neuron. All these things are found very commonly in experiments. Very simple circuit. But what you are going to see is that that extremely simple circuit of just 2 neurons, not even 100 billion, does remarkable things. We are going to put in just one additional small fact. The fact is that only these excitatory neurons get fatigued. So if you take out one excitatory neuron from the brain, inject current into it, make it active. There is no consciousness now, it's just one isolated neuron. That neuron is not going to be active throughout. The current will keep passing, and within a very short period of time, that neuron will stop reacting. But the amazing thing about the brain is, and this is again invariant across vast brain regions and species, is that the inhibitory neurons, most of them, not all, but majority of the inhibitory neurons don't get fatigued. Not at least on the short time scale of a couple of seconds. If you keep driving them for 1 hour, well, they're going to die. But on the short time scale of a couple of seconds, the inhibitory neurons don't get fatigued. So that's all we are going to put in. This is my simple harmonic oscillator, if you wish. Very little that we have put in. These are the equations. I'll very briefly describe them because we're going to solve them without going into the math. Nu is the firing rate of the excitatory neuron, nu E. Nu I is the firing rate of inhibitory neuron. C is the fatigue parameter. The firing rate of excitatory neurons decays with a function of time. There is some relaxation. It depends on the amount of excitatory input, and the excitatory input is where that fatigue is buried in. biology tells us that the fatigue comes into the synaptic connections or the something called dendrites of neurons, a beautiful topic for which I'm going to skip, and the amount of inhibition. Inhibitory neurons, of course, are driven by excitatory neuron and shut down by inhibitory neuron. They do not have that fatigue term, and the fatigue term itself eventually dies out. If you stop stimulating a neuron for a little while, it becomes perky and starts again becoming reactive. So this is all that goes into it, nothing more. You can write down on a small piece of paper, within a few months you can solve this. That's what it does. So in green is that fatigue parameter. The x-axis is time. Y-axis is the membrane potential. In blue is the membrane potential of inhibitory neuron. In red is the membrane potential of excitatory neuron. Notice I don't have to describe what this does. It shows you everything that our a lot of experimental data showed that we extracted. You get slow oscillations of the order of 1 second. You get very sharp transition to upstate. The upstates are very noisy, downstates are very quiet, and you get very slow transition to the downstate. So this very simple theory shows robust 1 Hertz oscillation, and this is scalable. I'm going to show you data for that. What happens when you go from 2 to millions of them? We can't do billions yet because we don't have the computational power, but already by the time you go to millions, you see something very remarkable, and we are going to get to that. It captures that. The up and down state oscillations are deterministic. This oscillation goes on indefinitely. But what goes on in the up state is actually chaotic. It's one of the very few physical systems that I know of, perhaps the only one that I know of, where you have coexistence of determinism and chaos. Sorry? This network can be scaled up as you increase the scaling, as you include more and more neurons. And this network already has plenty of stochasticity in it. As you increase the number of neurons, the stochasticity of the upstate increases, but the frequency remains rock solid, and the rest of the phenomena remain the same. The only condition in which the frequency of this oscillation changes is when out of all those parameters, 6 of them that I talked about, 1 of them is changed, namely the excitatory to inhibitory connection strength. If that connection strength increases, this network starts to do something very interestingly different, already different than what you see. And that's what this is. Once again, the x-axis is time. Y-axis is the membrane potential. Now, to simplify, only the membrane potential of the pyramidal neuron or the excitatory neuron. As it turns out, excitatory neurons are the majority in your brain. They take up about 80% of all the neurons. What you see, what's done here is that we have steadily, in this calculation, steadily increased the amount of driving force from the excitatory neuron to inhibitory neuron. And what you see is that steadily the frequency goes up, the amount of stochasticity goes up, and then the network makes a sudden transition when these oscillations are gone. And we believe that that is the state in which we are alert, perhaps. And I'm going to show you something about that. So at this point, I'm going to bifurcate and talk about 2 things. One is departure from this behavior in the brain itself. This kind of behavior is not seen in all parts of the brain, so departure is already interesting. Since we have captured this, we might as well look for departure. And second thing is this transition to the awake state. What exactly happens in experimental data? The theory has it. What about experiment? This was the prediction. We want to look at it. So first part first, what happens to the other parts of the brain? Roughly speaking, as far as the cortex goes, there are 2 parts to it. One is the new brain or the neocortex. Other is the old brain, also called as the limbic system, also known as the hippocampus, also known as the archicortex, also known as the Papez circuit, many names to it. That system is present even in reptiles, the hippocampal system. The neocortex is there, of course, in mammals. What happens is that in gray, what I have shown is the up and down state transition of the neocortex. especially the parietal cortex, which is the major source of input to hippocampus. In these three different colors are the responses of hippocampal neurons or activities of hippocampal neurons simultaneously done. So these are very difficult experiments, and the credit goes to brilliant students who carried out this experiment. This is published. That's a circuit right there. The neocortex is down below that projects to a very interesting region called the entorhinal cortex, which projects to the hippocampus. And in hippocampus, you have these three subregions, the dentate gyrus, where new neurons are born every single day in your adult life. That's the only part of the cortex when new neurons are being born constantly. Very interesting structure. If you interfere with the birth of those new neurons, you're not going to learn too much. Next is CA3, the most self-referential region of the brain. And third is the output of hippocampus called CA1. This region is extremely critical for learning, memory, and declarative memory especially. We are going to focus on these three, and what you see is this. The first step of the hippocampus, namely the dentate gyrus, shows you more or less the same slow oscillations, but of diminished amplitude. The next stage of hippocampus, namely the region CA3, those oscillations are completely gone. Not there at all. That's the standard deviation. And the output of the hippocampus, namely CA1, that is actually doing the opposite, like a push-pull. When your neocortex is going up, the CA1 is going down. When the neocortex goes down, the CA1 is actually becoming more active. So something remarkable is going on. The total activity levels in these parts are the same. If you had not looked at these individual neurons, and if you had not looked with millisecond accuracy, you won't have guessed this dynamic. In fact, if you looked at the inhibitory neurons in CA1, these are the excitatory neurons. Within CA1, if you looked at inhibitory neurons, they are doing the exact opposite. They're actually becoming more active. But in neocortex, the inhibitory and excitatory neurons are doing the same thing. What's the moral of the story? That even very simple, this elementary basic circuit of excitatory inhibitory neuron has enormous amount of richness. And it actually bears fruit in experimental data. Not only that, the most surprising thing happens, and these things are published, you can look at these things. The next thing is actually unpublished yet, which is this in-between stuff. So as I told you, in terms of evolutionary terms, the neocortex came the last. That's why it's the neocortex. Hippocampus is the archicortex, is the old brain. The region of the brain that connects the old brain to the new brain, that's called the paleocortex. It's also called the entorhinal cortex. Entorhinal cortex is a very mysterious structure. People started investigating that very intensely only recently, and it's been bearing enormous amount of interesting results, so many that I can't even begin to tell you about them. You find crystal structure in entorhinal cortex. Those neurons have amazing memory at the level of single neurons. We wanted to see what those neurons do when you're doing nothing to them, which nobody had done. And that's what you see here. This is too much jargon here, so ignore that. What you're going to see is this. You're going to see in this paleocortex, also known as entorhinal cortex, spontaneous, quantized, stochastic two-state dynamics. What do I mean by that? Let's focus just on this, because I'm running out of time, and I haven't even told you about what happens when we run, so I'll have to speed up. So in red now is the parietal cortical slow oscillation, the same thing that I showed you so far in many slides. Those neurons are oscillating away at about 1 hertz. That's in red. In blue now is the membrane potential of one very special neuron from the paleocortex. If you look at it broadly here, you will say, well, that too is showing you some same two-state oscillation, big deal. But this really difficult experiment, when you monitor these two brains simultaneously, tells you something remarkable. And once again, these two graduate students and postdoc really worked hard. It took us many, many years of effort to finally nail this down. And in the next slide, you will see why this gets so exciting. What you see is that immediately after the neocortex makes an up transition, here the paleocortex too makes an up transition. You can't see it, but believe me, it does that after merely 50 milliseconds. But then, when the neocortex goes to sleep, very briefly, just for half a second, the paleocortex just keeps chugging along. Neocortex comes up, this guy doesn't care. Comes up, doesn't care. It keeps chugging along, but suddenly, out of the blue, when neocortex goes down, the paleocortex also goes down, and so on and so forth. Here, it jumped across 3 states of neocortex. What is half a second? Sounds very small, but the currency in the brain is 1 millisecond. So half a second is a lot. In the next cycle, it went down for only 1 cycle. It went down immediately. In the next cycle, it went down after 2 cycles. So I'm going to wind up, and I promise, in 4 minutes. That's what you see. This is the activity of one entorhinal cortical neuron sorted. Some upstates in entorhinal cortex are short, some are long. And if you just looked at them, you'll see, well, there is some stochastic degree in terms of how long the upstates are. What this shows you is simultaneously what the neocortex is doing at the same time. The source of input, where you see these 2 oscillations, 2 states. What you see is that if this oscillation is long, it has either 2 or 3 or 4 or 5 neocortical states nested in it. And that's what is shown here. There is either 1 state or 2 state or 3 or 4. This is shown across 20, 30 neurons, thousands of amounts of data. Notice this is on log scale. You see a beautiful quantization of 1, 2, 3, 4, 5, and of course it is stochastic. So many of the things that you people might look for, this system is showing. Enormous amount of richness. Now, I'm not going to get into this, how this comes about, what's the theory of this. We can talk about it someday. I'm going to quickly end with what happens, the other prediction of the theory when you wake up. When you start becoming alert. And the best way to control that is, in this case, is to make the mouse run. This is the average EEG of the mouse brain. X-axis is frequency, y-axis is the power spectrum in decibels on log scale. There is nothing special going on. This is when the mouse has just stopped. He's just sitting around, like in the previous data. Is a bump here. This is the power spectrum when the mouse starts to run. Again, you don't see too much. Maybe there is more power, maybe less than that, but don't see it. All you do is you subtract them, which is tricky. These are logarithmic quantities, but that's more like division. When you do that, what you find is that actually, as soon as the mouse starts to run, the power doesn't go up everywhere. It goes up in 2 distinct bands. The slow oscillation, the slow gamma oscillations, which are between 20 to 40 hertz, and the fast gamma oscillation, which are between 40 to 90 hertz. 2 bumps, very clearly, highly significant. Same thing happens when you average this data across 12 mice, hundreds of hours of data, very clear appearance of 2 different bands of oscillation with behavior. We can now say, okay, this is pretty interesting. Incidentally, this gamma oscillation, tons of literature shows that in humans, the amplitude, the power of gamma oscillation correlates with how your performance is in several tasks. Here is the mouse's speed. The speed he's running now, this speed is 0, speed goes up, again he stops, runs, stops, and so on. This is the power in the slow and fast band. You can see it in the raw data. It goes up, comes down, goes up, comes down, and so on. This is the zoom in. That's a slow gamma, that's a fast gamma. Here is once again the data. X-axis now is speed, y-axis is amplitude. You can see 2 to 3-fold increase in the slow gamma oscillation. As the speed increases, the slow gamma actually increases progressively. It's not just all or none phenomenon. Highly significant result across all data. The shaded area shows the standard deviation, very tiny. Here is another interesting thing which we wouldn't have thought of. That's the speed dependence of fast gamma oscillation. It goes up, but that actually goes up logarithmically, while the slow gamma goes up linearly. We can solve this. I'm out of time, so I'm not going to tell you about this. There is actually even more interesting stuff happens, which is that the 2 oscillations that I told you about, the slow oscillation that we talked about in the first part, and the fast gamma oscillation that I talked about now, they are actually getting coupled. And the coupling between the phase of the slow oscillation and the amplitude of the fast oscillation depends on speed. And that's this picture, very interesting, which starts to get us into the immediate instantaneous level of what's going on. And I want to end here by thanking this large number of wonderful people in my laboratory, great collaborators, and the funding from these sources. And I'm sorry I ran over a little bit on time. Thank you.
I have maybe little bit technical questions. When you go for many neuron cases, people previously thought that the neurons which are passive, and there are two kinds of neurons which are very active and some are passive. Initially, people thought that these passive neurons, they don't take part in the activities. But then they did in vitro experiments, and they found if you block them, then situation changed radically. Now, in your mathematical model, how do you include these passive behaviors in the network model so that you can get a real picture? This is one. Second question, you are saying about stochasticity. In nonlinear dynamics, it is one of the very difficult issues that how do you distinguish the chaotic and stochastic structure from time series analysis?
Yes. So here is the activity of one pyramidal neuron. This neuron you will call active neuron.
Yes.
The amazing part, this is in vivo, not in vitro.
Yeah.
The interesting part is that this neuron fires, look at how many spikes it fires. When people say a neuron is active or not, it's based on the spikes or action potential. This neuron fired one action potential here, and things are going down. I'm running out of power here. Something is going on. This neuron fires one spike here. At most, it fires one spike in 10 seconds. So the remarkable fact about the brain is that majority of the neurons are completely silent.
Yeah, but they take part actively,
even
though passive.
That's right. Even this neuron that is actively taking part is firing only one spike out of 10 upstates.
Yeah.
So that's actually an interesting point in itself. That why are the neurons in the brain so silent is the opposite point of this. On one hand, neurons are active even when we are anesthetized. But on the other hand, when neurons are active, they're active so rarely. And this is true not just when they are anesthetized, even when the animals are behaving. We have done these experiments, we have seen that when the animal is behaving, some neuron, no matter what you do, is not going to fire a single spike. Whereas the other neurons which are going to respond, they are going to respond at most on an average same number of spikes, about 1 per second.
But you didn't do mathematical modeling.
We have done that.
No, for many neurons.
We can do that, but what I'm trying to tell you is that the amount of firing rate of these neurons is so low that differentiating between active and inactive is a moot point. So this guy fires one spike in 10 cycles, which means it's pretty much inactive in itself.
Okay.
And it's beautiful work. Thanks for sharing it. About the simple model again, a quick question and then a follow-up. The quick question is, does it display hysteresis? So if you go down, the transition points at a different time. And I'm thinking, that's interesting. And then I'm thinking how that relates to existing models of anesthesia, that a lot of these anesthetic compounds, such as propofol, act on GABA receptors.
Yes.
And is that consistent with your model then in explaining when and why the transition occurs in terms of these pharmacological effects of anesthetics?
Yeah, great question. So as you can see, first point, the two-state model, stability, and so on, you can see that here. The stochasticity, as well as the previous question that I could not answer fully, you can see that goes on here as well.
But that's not showing hysteresis.
Hysteresis is actually because of this parameter, the C parameter. Hysteresis is because of this parameter.
Okay.
What does C do? Hysteresis is in this single excitated neuron. If you make it active, it's going to start becoming silent. That's your hysteresis. It's buried into it. So that tiny little bit of memory of the recent activity is there in the excitated neuron, not in the inhibitory neurons. And that's the crucial parameter which is generating this. Notice that's the hysteresis parameter which is actually out of phase with this two-state oscillation. So that's what is doing it.
Okay.
And it's due to that hysteresis that your up transitions are sharp, but the down transitions are slow.
Yeah, no, it wasn't that hysteresis. I was thinking as you change the excitatory drive, that you showed the transition to a fully active state.
Right.
And it was hysteresis in that transition that I was interested in.
That's a very interesting question. So it turns out that brains, individual neurons and synapses, as we discussed, show hysteresis on multiple time scales. What we have barely scratched here is one type of hysteresis alone. And once we start playing with other kinds of hysteresis, such as adaptation, long-term adaptation, synaptic facilitation and depression, then these different anesthetics you're talking about, that comes into picture. And that's where we will get to, but sure.
This is a very short comment. Your coexistence of determinism and chaos has an extremely interesting history going back to work in MIT in the 1960s and 70s. Stuart Kaufman has built an entire career around the concept of the edge of chaos, which was actually refined by the Danish physicist Per Bach into using his concept of self-organized criticality into the criticality condition. Now, the criticality condition is known to pervade physiological systems because it gives rise to the grounds for fractal responses in the physiology, which is most famous in heart rate variability. Now, this is a universally acknowledged condition for health. It is also a prima facie condition for the coupling of consciousness in a way that I could show you later. But this is an extraordinarily important point that you've brought up for actually coupling consciousness into a physical system. I thought it was a beautiful talk, and I really congratulate you on it. Thank you.
Absolutely. And the interesting part here is that the system actually goes in and out of chaotic state. That's what it's doing. That's very surprising. And that part in itself is generating all these subsequent dynamics. All this is because of the upstate. I really thank you for bringing out that point. Notice the interesting thing is going on the transition to the upstate. That's when you see this very interesting dynamic in the old brain, and especially in the paleo brain. And I think that there is that part that I really ran over. There is something really amazing going on there. But we'll go there later. Yeah.
There's one question about the neurochemical transmitters. When we say this excitatory and inhibitory neurons, but if you give excess of the stimulatory neurotransmitters or inhibitory, which we see in clinical condition.
Yes.
Now, what balance would be there? Whether the same balance would be there or it will be disturbed?
Right here.
Yes.
So the key parameter is this. So that's a network right there. That's a network. Those are the parameters. The parameters, if they go completely out of bound, nothing happens. Either basically the network just dies. And it's interesting, not just if there is too much inhibition, that's when there's nothing happens, but in this version, even if there is too much excitation, the network is going to die. Because now the excited neurons are going to be perpetually fatigued, a large fraction of them. And that will do that. But the interesting part is here, that one key parameter, that's the unique prediction of this theory, that out of all these parameters, the driving force from the excitatory neuron to the inhibitory neuron. That's the parameter that determines this frequency, none of the other parameters. And this is something, even in this very simple model, we didn't predict when we started. We had to actually solve it to get to this knowledge. So we have various amounts of pharmacological tools, which, as you know, we can hit either GABA or different receptors of GABA, and especially different receptors of the glutamate, whether they are voltage-gated, ligand-gated, and so on. And those different guys target different kinds of connections. And that's why we'll be able to test this experimentally.
One very small thing, like we see normally people take alcohol.
Yeah.
And some people get very excited, others don't get excited.
I knew that. Maybe after a beer I'll know.
Okay, I think one is very nice. I don't have a question, we can stop.
Thank you.
Speaker.